growth curve of cells. So for example, if you get some cells in that you've never worked with before, then yeah, you can go by the data that's in the handout with the cells. But what you're going to do is you're going to do a growth curve to find out how fast these cells grow with your hands. Okay, so in your lab, you're going to find out how fast they grow. So what you have to do is you have to set up a growth curve. Now that's what you're going to be doing in this experiment, is setting up a growth curve. You're also going to learn how to test for cell viability. There's a solution up there behind you. Just turn the camera around and point straight up, and you see those two little bottles that are all blue? That say right there, Trypan Blue?
Oh, yeah.
Okay, so there's two of those bottles, and those are going to be our solution of Trypan Blue that I'll show you how that actually works. And we'll use that to test for cell viability using the hemocytometer. But you're going to draw out a growth curve, and you're going to determine the doubling time for your cell line. And I know approximately what the doubling time needs to be. All right, now you're going to use the same cells that you're working with before. You're going to use the same media, and you're going to use the same trypsin. However, if you'll note on number 2, it says you will also need the following setups for working outside of the hood. So there's going to be a part of this experiment that you're going to do without having to do it in the hood. Okay? It's going to be the counting of the cells to find out how many cells have grown. So you need to set up 2 8 mil aliquots of trypsin EDTA. So you need 2 15 mil tubes that you have to pull out of here and put in the refrigerator with your name on them. They do not, and from that point on, you just do not consider them to be sterile, because you don't need them to be sterile. You're doing everything outside the hood. Because the only time you need sterility is when you have live cells that have to be the results. In this case, you're not going to be wanting live cells. You're only wanting cell numbers, and then you throw them away. Okay? You also need, set up with your media here, you need to get a 15 mil tube out and put 14 mils of just media in there. Put your name on it and put that next to your trypsin EDTA. And we'll talk about that in a little while when I talk about how you count them. You'll need 35 millimeter plates. These 35 millimeter plates are around there above the microscope, top left. You know, take it over. It should be top left up there at the top. Okay? They look about this size. They're just standard tissue culture plates. I'm only going to, it says you need 12 of these plates. I'm only going to do 2 today because I don't want to waste them. But you're going to do 12 plates of these.
How many are in total?
I think there's 20. Let me check. Yeah, 20 per bag. So I'll do 4. That way, you know, you can at least see how these things work. So what we need, this opens up just like the other ones. You'll notice here it shows you where to pull it open. So you pull it open, get it opened up, and keeping it, you always pull it from the correct side. Open it up, keeping it held down. I typically pull out 2 at a time. I don't want them to pop open. They're just little bitty tissue culture plates. So there's 4 of them. And keeping it tight, just like before, I'll tighten it down and tape it up. Anybody having problems with, by the way, is anybody having any problems with their tissue culture? And it goes back inside here.
No.
You are?
I don't know, I'll check on it.
Okay, all right. Okay, good. All right. You're also going to need a hemocytometer, and you need to clean it with 70% ethanol ahead of time. Be sure to do it over there on that diaper. The 0.4% Trypan Blue, which is behind you over there by the above the water bath. That's the ones I just showed you. You need your pipettes. You need 15 mil tube and a 1.5 mils microcentrifuge tube. The 1.5 mil microcentrifuge tube, if you'll look here, this is it right here. The way some of you already know this because you've taken molecular biology lab, but the way you get tubes out of this, you do not reach in. You shake them out. So you just open it up. And you shake out a tube. And you close it back up and put it back over there in the corner. I have this nice little holder here. And you put it there, okay? We'll do that. We'll use that when we do the counting of the initial counting of the cells. Traditional tissue culture plates. So we need one 10-centimeter plate in this procedure. Now note, by the way, these are 35-millimeter plates. Y'all see this? These are 60-millimeter plates. They're different in size. Notice the difference. These are 100-millimeter plates, 10-centimeter plates. Okay? When in doubt, read it. Make sure. All right. Good job on taping this all up, guys. So I'm going to pull one out. Alright, so we got that. Alright, so we go to the procedure. Let's read through the procedure first. You'll notice it says the following procedures 1 through 11 must be done in the hood. So this is where you're setting up your experiment to incubate for almost 12 days, 6 days. So that means you want it to be sterile, okay? Because you don't want to have yeast or anything like that interfering with it. I've already warmed up my media, just like you standardly do, and the trypsin. I've already got it in the hood after washing it with 70% ethanol. Now I've got my cells in there, and I'm going to suck off the media off the cells using the vacuum line. So let's go ahead and turn the vacuum line system on. I need to put some bleach in that. Then we're going to add 4 mils of the trypsin. Incubate for 5 to 10 minutes. While the cells are incubating, we're going to put 10 mils of MEM in 15 mil tube and in the 10-centimeter tissue culture plate. After the cells have lifted from the plate, transfer the 4 mils of cells to the 15 mil tube. Before transferring trypsin, suck up the trypsin and gently expel it back on the whole surface of the plate. So you want to make sure this is one particular situation where you want to make sure that you have gotten nearly every cell off the plate. You want to try to have cell numbers that you're lifting cells off plates that are between 70 and 80% confluent, because you're going to need a lot of cells. So if you can't find one plate that's 70 to 80% confluent, but you have 2 plates which are 40% confluent, use 2 plates still instead. Okay? Because then what you'll do is just combine the cells together. So what you can do is spin them in 2 tubes, and then when you resuspend them in 10 mils, you resuspend 1 tube, transfer that to the next tube that has the other cells, and resuspend that, and you've combined the cells. Does that make sense? So that's one of the things you can do if you need to do that. If they're 20% confluent and there's enough plates in there, then take 3 plates and do the same thing, or take 4 plates, do the same thing, because you can resuspend them together by just moving the 10 mils tube to tube to tube. All right. All right. So you centrifuge. You rinse the tube after 70% ethanol after you centrifuge. So all of this you pretty much know, right? It says carefully suck off the soup and resuspend the cells with 8 mils of MEM. All right, so it's 8 mils times 10. Now you're going to immediately split the cells 1 to 8. Now the reason you're going to do this is next Wednesday, while this experiment is going on, I'm going to demonstrate cryopreservation to you. So you're going to be wanting some cells to do the cryopreservation experiment, while you're doing these countings for like 6 days, so you can get this next experiment out of the way. All right? And in that one, that's a 2-part experiment. In that one, you first have to put cells down in a frozen state. Then 3 to 5 days later, you have to come back and bring them back up out of frozen state, and then take pictures of them growing the next few days after that. So it's a 2-part experiment demonstrating that technique to you. So you need to have some cells in this first part. So now you've got 7 mils of cells left. That you put 1 mil into the 10 centimeter plate. Now you've got 7 mils of cells left. So gently vortex the remaining 7 mils to distribute the cells evenly. Aseptically remove 160 microliters of cells from the tube and place it into this 1.5 mil tube here. Okay? Now you're going to add 40 microliters of the Trypan Blue. Make sure you stir the Trypan Blue up. So sit there and stir it, because you want to make sure it's evenly stirred up and in suspension. Then suck up 40 microliters. Place it on your cells and gently mix. Y'all remember how to mix a 1.5 mil tube like this? By hitting it like this. Okay? So now, since you've got it in here and these cells are not going to be used again, you can go up there and you can do the actual placing of your cells into the hemacytometer without doing it over here. You want to move fast in this project. You want to get the cells counted as fast as possible. So what you can do, I'll have some pipettes over there. And what you can do is go up there, mix it up again, remove, in this case 10 microliters as you read here, a stained cell combination to each wedge. So you want to put it on both sides, okay, on the cover slips, and wait 1 minute. By capillary action, the media cells will be drawn up under the cover slip. And what's happening here is the Trypan Blue, if you read down further, remove the hemacytometer from the hood and count cells with microscope. The procedure for counting will be covered in the lab below. Observe the cells with low power microscopy and count the total number of cells, stained and unstained. And then count the number of stained cells. These unstained cells are the viable cells. And then you'll compute the concentration of viable cells per mil. What happens here is the Trypan Blue readily crosses the membrane of dead cells and living cells. But living cells pump it back out. And so you'll have a blue background with a white cell. That's a living cell. If it's a dead cell, it'll be a blue background with a blue cell. That's a dead cell. So this is called viability test. All right? So it's a Trypan Blue significantly overestimates the number of viable cells, but it's sufficient for the purposes of this lab. However, because of the overestimate, we normally assume that approximately 30% of the cells measured as viable with Trypan Blue will not be able to continue growth beyond 24-hour period. So we have to include that in our calculations, that 30%. You'll count the 4 corners on both sets. So it'll be 16 square sections. You'll count 4 over here and 4 over here. And then you divide by 4. So you count 4 over here, 4 over here, and you divide by 8. Excuse me, correct that. All right? You really only need to count 1 set of 4. The reason you do both of them is just in case this one looks weird, so that you don't have to do it again. So you really only have to count 1 set. Then you multiply that number by 0.7. What that does is it eliminates 30% of the cells that we're assuming are going to be dead in 24 hours. Because remember, we're trying to plate out cells that we're going to count over a 6-day period. So we want to have the number of viable cells to start with as our baseline, as 0.0. What we start with is what we want. And then by 10.4 to get the number of viable cells per mil. Since you're counting the number of cells in a 1.45 mil tube, that has 160 microliters over 200 microliters, we've done a 4 to 5 dilution of the rich cells. You will need to divide the number of viable cells by 0.8. So you have to make that change there to obtain the true number of viable cells per mil in the 15 mil tube. For example, 31, 34, 35, 32 equals 132. Divided by 4 is 33. 33 times 0.7 times 10 to the fourth is 231,000 viable cells per mil in the 1.5 mil microfuge tube. Divide that by 0.8, we actually have 288,750 viable cells per mil in the 15 mil tube. Oh no, just divide by 4. The reason I'm saying that, the reason you do it 2 sets of those, in reality, I now remember, is just in case this one screws up, you at least have one working. So you really only need to count one of the grids. 4 16 cell grids. The other one's your backup. It's always nice to have a backup. Okay? It really is on this. All right. Now, in order to determine that we have enough cells to distribute 50,000 cells into each 35 millimeter plate, we will calculate the total number of cells remaining in that 7 mils. So we'll take our 288,750 viable cells per mil and multiply it by 7. That tells us we have 2 million cells. We divide that 2 million by 50,000 cells to see if we have enough cells for 12 plates, and it tells us we have 40 plates. So yes, we do. All right? Now keep this in mind. If you don't, let's say you only have enough for, let's say your number is far much lower than that, and you only have enough 50,000 cells for 10 plates, but you need 12 plates. Then to reduce the number of cells you start off with to 25,000. Okay? 50,000 is just an ideal number, but you can go less. Just as long as you know what that number is that you're starting off with. Okay? This is when you need to write down all your calculations. Keep track of what you've done and what you did your calculations on. Alright? So, we next determine the volume of cells we need to add 50,000 cells per plate. So 50,000 is divided by 288,750 cells per mil, which ends up being 0.173 mils, which is rounded off, equals 173 microliters per plate that we add. As I said before, if you do not have enough cells, plate out somewhere between 25,000 to 50,000 cells to work with. Do y'all understand up to this point? All right. I think the initial thing I need to show you is how to do the Trypan Blue stuff, as well as to put the cells in here. So that's what we're going to demonstrate right now. Okay, can you hand me some pasture pipettes over there? We ran out. Give me one that has something in it. Oh. Put that one over there in the used. Put that other one in the used thing over there.
To your right? Right there.
Yeah, they're right there. Used pasture. I wonder how that happened. Yeah, one left. Usually I fill them up. All right. Go ahead and take my ring off. Tell me what's on it. I'm not talking about this right now because this is something I've already demonstrated, right? turned you on. Oh. Be sure to look at your cells before you work with them. I've already looked at these cells. Can you put this in the incubator? I've already looked at those cells, so I know that they're in a pretty good state to be able to do everything. I feel like I'm acting. I keep waiting for somebody to say cut. All right. Okay, so you got about 5 to 10 minutes on those cells. One thing you can do, somebody read for me how many oh, look at that, it's all messed up. Somebody read for me how many mils of media goes onto the 35 millimeter plates. Is it 2 mils? I believe it is. Amount of media in the plates?
No.
Seriously. That is weird. Okay. So where it says, are we missing somebody here?
You should, analyze.
Here it is, right here. Following procedures, number 12. To each marked 35 millimeter plate, add 1 mil of MEM. MEM, okay? So I'm gonna go ahead, I'm gonna be adding 10 mils to this plate, and I'm gonna be adding 10 mils to a tube here. And while those cells are incubating during this short period of time, I'm going to go ahead and have them add the media to them. So here we go. So you're going to have 12 of those little 35 millimeter plates. So you'll have about 12 of these. What I normally do is give them, stack them up 2 at a time, and so you can just line them up like this. And this will go up to 12. You'll notice here it goes up to 12. So you can go all the way up to 12 like that. Now since I only need 2, I need 4. I'm just taking it up to 4. See how that looks. Okay. As long as they don't get too high, they're fine. You can do this if you want to get rid of a bubble. All right. So we'll put those back there. We could put 2 mils in there, but those plates, when you start moving them around and you put 2 mils in them, that media has a tendency to slosh a lot. So when you put cells in them, And initially kind of dribble them around the plate and then gently swirl them so that you get a good spread across them because you don't want them to become confluent. Okay. Can you hand me those cells that are over there? So I'll have all my plates ready and everything's ready to go. Thank you. So just look at this, you can see the cells coming off here. Actually, look here. See them?
It's yeast.
It's not yeast, I already looked at them a little while ago. So what I gotta do is just make sure I break them off here. But these things were like 80-something% confluent when I got them. You'll know yeast when you see yeast. Alright, so since I'm going to be throwing this plate away, I'll just put the lid over here and just work like this. Now this is when you want to do some squirting because you want to get as many cells off there as possible.
Yeah, she left.
Are we missing somebody?
Anne left to go to the thing.
The thing? What thing?
Remember she was talking about it like last week.
Oh.
Some.
Oh, yeah.
The physics thing.
Physics thing, okay. Alright, so there we go. I need to spin it for 3 minutes, please. You didn't know you were going to become my TA? That one should be already clean.
Are you gonna go? Are you gonna go?
Confluency is always estimated.
Oh, it's completely estimated. You base it upon areas, looking at multiple areas under the microscope. And I mean, if there's 50% clear space and 50% cells, that's pretty straightforward. After that, below that and above that, you're pretty much estimating. All right. So I need to use 8 mils of cells, is that correct?
8 minutes.
I believe it was.
Yes.
All right.
You do that more.
So I'm resuspending the cells. And I'm taking 1 mil immediately and putting it in here, swirling it, I'll put my name and the date and the name of the cells on there in a little while. So now I got to take this guy. I'm going to take 160 microliters. So I'll turn this to 160. Unfortunately, they discontinued these, which really drives me up a wall. They're so much lighter than Rannon's and so much easier to work with, but they discontinued them. They got bought out. All right, so stir it a little bit. Get it resuspended. And then get in there and take you 160 microliters real quick. Add it to here. Can you hand me the Trypan Blue over there? Thank you. Stir it. This one you want to wear gloves.
Can we use the stuff and genetics as well?
Probably, well, no, I think you use something else. But you do want to wear gloves with this. I'm less likely to spill on myself than you guys are. All right, we're ready to go. So we move quickly over here. Can you bring me a P20 out there, please? So we got this one's already clean, it looks like. Oh, it doesn't look like. Where's the cover slip on it? Oh, okay. Yeah, I'll use that. All right, so I'm gonna go ahead and set this up. Find a P20?
Trust me.
As I said before, y'all know that if your cover slip breaks, I got extras here. All right, so we needed at 10 microliters. I'll have some of these set right here, like this. They'll be sitting here just like that you can work with. So I'm going to do 10 microliters on each side. Stir it up. See the blue color? So they've probably been soaking for about a minute, so they should be ready. So as before, get your find the little arrow. There's the arrow. I lost it. All right, let me find it first, and then I'll. Okay, now I've got the arrow, and I'm going to go to the grid. This on what? I thought it was on 20. No wonder I was having problems. Try that again. That's 40x. Okay, there we go. Alright, so I've worked pretty good here. Let me show you what. I don't have very many dead cells. Why is that zoomed out? Is that zoomed? Because that doesn't look right.
It looks zoomed in.
I mean zoomed in.
And the colors.
I'm trying to see if there's a zoom on this, because I don't think there was one. But if you look at these, there's very few dead cells. When I look at them underneath here, take a look. I don't see any dead cells. And that 116 square.
Yeah, they're on the side.
Yeah. I'll look around for a few moments and see if I can find a dead one. You want to take a look?
Yeah.
Jose? You can videotape that, I bet, inside there. Yeah. All right, I'm gonna look around and see if I can find a dead one, just so y'all can see the difference in them. Okay.
Is that one?
Yeah.
You see him? See the dead one there?
Yeah.
See the difference between them?
Are those one other ones in the background nearby?
No, those aren't. Those you talking about these guys right here?
Yeah.
No, those aren't real cells. Those are just fragments.
Okay.
So that's a dead cell. I'm just looking around for some more. There's another one right here. Now sometimes you'll have a bunch, sometimes you'll have a few. So in this particular case, we're in pretty good shape with counting. So I'm going to start counting. So I like starting off at 0, so I'm going to count real quick. Okay, that's 56 for that one. There's a fun one. So I'm counting all the cells that I see, but I see a blue one down here in the corner. Look here. See that? So I'm going to count all of those, but I got to count, I got to record how many were dead also.
Which one's the dead one? Like this one?
No, here, let me show you.
Right there.
Right there, there's one next to that one, there's dead.
That one. Yeah, that one. Okay. I got you.
So would you use like the last clicker to count that one?
I was planning on it. So what I'll do is, and then I'd have to take the divide that by 4. But let's see if we can do this. Okay, hold on. Let me count all the cells now. Oh, there's another one dead. Okay. All right. Okay. Then that still adds on to your stuff, so you got to be careful with that one.
Yeah.
Right, so here, now I got this weird thing down here, but I'll work around that. Okay. You see any dead ones there? Looks like a dead one in there. Okay, I think I see one dead one here. All right, so I have 224 minus 3 dead ones. So that gives me 221 divided by 4. That should be 55, or around 55 cells, and total. And then divide 3 by 4, so that's 1. So it's 55 minus 1, so that gives me around 54 cells that are alive in the current thing. So I got that number to work with. Okay, does that make sense? Mm-hmm. So you're not going to have a whole lot of dead cells in this first group unless you have really messed up.
Yeah, I really think I have. So.
Okay, so but they should look fine. All right, so you get it back down to 0 when you're done, and turn your light off, and then clean it. Record your data before you clean it, right? You don't want to sit there and have to redo it again, right? So make sure you record your data. Spray it clean here on this diaper, so that you don't drop it. Don't bother worrying about getting the diaper dirty. That's why they're called diapers. If you haven't changed a diaper, which is a rare occurrence for me. I did do it once in my life and had to be reminded that I actually did it because it was the most horrifying thing in my life. Oh, there it is right there. I had to change my niece's diaper when my brother and sister said they would be back in time. No, they didn't show up in time. So my nephew, he was like, "You're recording this, aren't you?" I'm not going to say anymore.
Good thing you caught yourself.
Yeah. So we have to take in consideration that 30% of those are going to be dead. So we take 54 times 0.7, excuse me. Let me redo that. 54 times 0.7. That tells us that we have 37.8 cells. And we multiply that times 10,000. Okay, so we're in a pretty good range, which is about equivalent. So 378,000 cells are viable in that 1.5 mil tube. So that's the concentration. However, since we diluted these, we need to do a dilution factor in there. So I take, what happened to my number? It was 37, I said 378, right? Mm-hmm. 378,000. So we have taken into consideration a dilution factor, so we divide by 0.8. So in the actual 15 mil tube over here, I have 472,500 cells per mil. Now we go to the next step. I'm sure I'm going to have enough cells, but I want you to do this just in case. I want to see the calculations in your book. So we multiply that times 7. That gives me over 3 million cells. I divide that by, let's see, I divide that by 50,000. That means I can actually do 66 plates. All right, so times 50,000 divided by 7. So I need to find out what concentration or what volume do I need to get 50,000 cells. So I take, I hate this thing right here. I take 50,000 divided by 472.5. So that tells me I take 106 microliters of cells. Okay? And you write that all down. So I've got that down. I've come over here. It's been a short period of time. And I've moved this to 160 microliters, which I've already got it set at. It was 106, wasn't it? So I set it at 106. I go ahead and stir it up a little bit, trying not to touch the top. If I have to, I'll use a pipette to stir it up if I feel like I need to, which it does look like it. So I'll take a 5 mil pipette. Yeah. So take a 5 mil pipette, get it completely stirred up again, and working very fast. Go in and get 106 microliters. Slowly swirl it, set it aside. Take this, do a gentle thing like this with it, keeping it stirred up. Go in, get the next one. Get the next two, next one. They hit bubbles at the top, so sometimes you gotta pop the bubble before you go in. Stir that one up. Do that with the remaining 4 sets. Then you can throw these cells away. You're done with them. I would mark this is set 1, set 2, set 3, set 4, set 5, set 6. When you place them in the incubator, place them in a row so that when you pull them out, you pull them out and then go over there and use your trypsin EDTA that you have in the refrigerator and your media, and we'll talk about that next level here. So you'll do this, do that all over there while these are incubating. But if you keep them in a row, when people reach in there, because everybody else will be in a row, they won't hit yours and knock them over. Does that make sense? All right, so let's go to this next level here. So I just talked about this. Procedures 13 and 16 should be, oh, here. So, day 0 is going to be today. So the number of cells that we plated on the plate on day 0, we put approximately 50,000 cells on the plate. Now these 35 millimeter plates have a surface area that is 8 square centimeters. You're going to be representing how many cells per square centimeter. So you divide 50,000 by 8 square centimeters, and that number comes out, according to this, 6,250 cells per square centimeter. Okay? You will plot that versus times 0 in your graph. Use a 3-cycle semi-log paper. So the y-coordinate is going to be the log. The x-coordinate is going to be the regular numbers. Okay? So that'll be your time, and this will be your number of cells. So it'll be a number of cells per square centimeter. So you'll plot, you'll go from 0 to 10,000 to 100,000 to 1 million. So those will be your 3 cycles. 0, 1,000, then another 10 would be 10,000.
Then 100,000?
Oh, wait a minute, let me do that again. Okay, 0 to 10,000, 10 to 100,000, then 100,000 to 1 million. Okay? So each one of those logs is going to be a 10, a factor of 10. Alright, so that, remember that. Everything you're plotting out is in cells per square centimeter. So when you count these cells, you're going to count 2 plates at a time. So that means you're going to use the hemoscytometer. You're going to count, one plate's going to be on one side of the hemoscytometer, the other plate's going to be the other. You are not interested in viability. All you're interested in is total cell number. And when I say total cell number, you're looking for total cell number divided by 8 centimeters. All right, so the cells in the 2 plates marked day 1 are counted at a minimum of 24 hours later. So if it does not have, and so what, for example, you could plate your cells out on Friday, come in on Saturday, that will give you your downtime on the cells, and then you come in on Monday, that'll be going into the actual exponential growth. And if you count every day after that, you will have this nice, beautiful exponential growth line.
About Friday to Sunday.
You can do that if you want to. All right.
Busy Saturday, so.
All right. So, just keep track of the time since you plated the cells. The important thing is record the time when you put these cells into the actual incubator. Record that time. And every time you count, record that time. The moment you split the cells, record that time. Okay? And that's what you're going to be plating these cells out according to that. Not days, but time, hours, number of hours. All right, you'll be graphing the number of cells per square centimeter versus the number of hours since plating. Remove the media using a pacifier. I'll have a little bulb up there that you can use to suck off the media. In fact, I have them right here. Come over here, guys. See this? So you just suck off the media and spit it down in here. Okay? Don't throw these away. Keep those. All right. You remove the media with the bulb, dispose of the media in a waste beaker, and then add 0.5 mils of your trypsin EDTA that you have in your refrigerator with your name to each plate. Then add and incubate for 5 minutes. After 5 minutes, use a P1000 pipette to suck up the TE and squirt over the plate surface 3 times. So what you're doing is you're trying to break the cells up so they're individual cells. So you'll just keep doing that. Once you finish that, then you'll get a fresh tip and add 0.5 mils of the MEM that you have in there to kill the reaction. Mix the cells using the pipette, the next ones that you add to it. Remove from each plate 20 microliter cells and count them. So put 20 microliters here, 20 microliters here. Now you've got 2 plates, you count both sides. You take the average of both sides. And then you take the average of those 2. Okay? So you can take all 8 counts and divide it by 8 if you wanted to. And that will tell you what you have in numbers. Okay? So then when you plate it out, you actually are plating out the counting. Your average is the average of 2 plates. Does that make sense? And I try to break it up here. All right. We are not staining the cells. We want to know the total number of cells that were produced up to this time point. Again, make sure the cells are suspended before adding them to the hemocytometer. Otherwise, the cells would settle. Record the total number of cells that are in your 1 mil of media and TE mix for each plate. Divide that by 8 centimeters, which will equal the total number of cells per square centimeter. Since you're growing 2 plates per day, take the average of the 2 plates for cells per square centimeter. This is the number you will plot versus hours collected on the growth curve. Repeat these steps for day 2, day 3, day 4, day 5. Doesn't mean it has to be exactly 24 hours. What I'm just saying is on the 2nd day, 3rd day, 4th day, just record the time so that you can plot it out correctly. So if you do this on a regular basis, you should see a plot that has a lag period, and then a lag period will start to come up, and now they'll start to go to exponential growth. So remember those, we plated out 50,000, we're assuming that 30% of them are going to die, or at least we tried to assume. Well, they're going to take a little time to recover because they got to rebuild their cell membranes because we just destroyed their cell membranes. And then they got to come back up and start growing and exponentially. So, using a 3-cycle semi-log paper, label the x-axis as hours since plating and the y-axis as the number of cells times 10 to the 4th minus 4th per cell square centimeter. Refer to figure 20.8 and section 20.9.2 of the book. If your day 0 is this number, 6,250,000, unless you did less than 50,000, it could be less. Mark the first cycle, y-coordinate, as 0 to 10,000, then the second cycle, 10,000 to 100,000, and the third cycle, 100,000 to 1,000,000. The x-coordinate will be linear and in hours since plating. Okay, well this is easy, you can do this. So day 0, Placed on the Y coordinate, it's time 0, so it's very much at the corner, up a little bit. So for example, if it's on a 3 cycle, you'll count, this is 10,000, so you'll count up. If it's a 6,000 one, you'll go 1,000, 2,000, 3,000, 4,000, 5,000, 6,000, and you try to rough it out, and you mark that right there on the Y coordinate as time 0. And so that's your beginning point. Once you actually have information done, you're going to actually figure out the population doubling time. That's how fast the cells double. I know approximately what it should be. So what you'll do is, using points on the exponential growth curve, part of the curve, you'll choose one at time, let's say 50 hours, and you'll go up and you'll find that point on the curve and record the number of cells that are recorded according to that curve as being there at that point. Then you'll take something like 75 hours, if it's still on the exponential growth, and you'll record how many cells there. So those are your 2 time points on the exponential growth. Then you'll take this value, t represents the time interval between xb and xe. So the, b is the cell number at the beginning of the selected incubation time, and e is the cell number at the end. So that's that second time point. So you'll take that time, divide it, and multiply it by the natural log of 2. These are just the values that they came up with to help in doing this population doubling time. Then you'll take that divided by the natural log of xe divided by xb, and then that'll give you the number of hours that you're working with here. Okay? How many hours does it take to double the number of cells? That's what it's doing. All right? Straightforward? All right, just take your time. You'll see. And this is a good experience because this is when you really have to do the counting a lot. Now remember, you don't have to do the counting. Once you got them plated out, you don't have to do the counting in the hood. The only thing you need to go back to the hood is to make sure you got cells for the next experiment. So still do that. All right? Okay, cool, guys. That's it. It's time to go home.